So good morning. Today we'll be doing a virtual lab. This lab is bacteria identification, where we'll be using different steps such as PCR amplification, PCR purification, sequencing prep, until we could analyze and identify a bacterium. And we'll be doing this for different set of samples, ranging from sample A all the way to sample F. So, since these steps are similar to those from EA all the way to F, I'll only be recording and showing the first set, which is for the sample A. Then afterward, for sample B all the way to F, I will just be showing the identification of each of the bacteria. So let's start off with the first one. As it says here, we need to do a sample preparation, and according to this bacteria lab, we should put on rubber glove. Then after this, we'll be obtaining a culture dish. Now we click on the wire loop to continue. Click on the bacteria colony here so we could grab the bacteria colony. Then we'll be click on the microfuge to transfer the bacteria colony from the wire loop to the micro centrifuge tube. Now we'll be adding digestive enzyme to the sample. So here we know that we have to use the pipette and discard the tip. Now we need to let this sit for a few hours, well, in real time, but this is a recording. Now we click the centrifuge. And it stays here for a period of about 3 hours. Now, after enzymatic digestion has occurred after several hours, this sample is ready. Now we move the tube into the hot water bath and we just place it in there for a certain amount of time. Now it is denatured and inactive, and now we need to click on the microfuge tube to continue the process. Since after this has been placed in the hot water bath, then it is placed into the centrifuge. It is centrifuged for amount of time as well. After it is complete, we unload the centrifuge. Unwanted debris has now been spun down. The DNA we want is in the superend. So now we click on the microfuge tube to continue. Transferring the superend to the PCR tube. The sample preparation is now complete. Click the orange tray to continue the part 2. Part 2 is the PCR amplification. Now here for part 2 it says PCR amplification, and on the right hand side it gives a description of the steps we need to do and what it is involved in each step. Now we add PCR master mix into each of the test tube, in each of the tubes. Now we add a positive control. Now for the negative control reaction we add deionized water. After that, we click the PCR machine in order to open it up and load the PCR tubes. After they have been successfully loaded, the PCR is closed. Here is animation of what is going on inside the PCR machine. Here it says that each for each cycle there is a number of DNA copies, and as we see as the cycle go on, the number of copies actually increase. So after this, we click done. Now the PCR's amplification is complete. Click the orange tree to continue. Now we move on into the next step, which is PCR purification. Now we do click the yellow buffer solution, and we add this buffer into the tubes. Now we click the orange tree to continue. Now the tubes containing PCR sample, positive control and negative control, will be put it on ice. So now we continue by clicking the orange tree. Now we load this one into the centrifuge. Now we click on the centrifuge to continue and leave it there for approximately, well, in this simulation it says 15 minutes. Now that it's trapped in the column, we need to loosen it up, and now we add a new collection where the column is inverted, and we add buffer solution to the inverted column. The first collection tube is no longer needed, so we click the orange tray to discard of it. Now we'll assemble to collect DNA into the collection tube, and we place it back again into the centrifuge. We leave it there for approximately 2 minutes, and then we unload it out of the centrifuge and click on the orange tube to we discard the column. The PCR product has been verified. Click the orange tube, continue. Now we move on to part 4, which is sequencing prep. Now here we prepared for sequencing. First, we'll dilute the PCR product to increase the volume by adding distilled water to the purified PCR product. Now we add the PCR product to the strip tubes. After that, we do the PCR to continue, and we load the strip tubes into it. We click on the PCR machine to continue. This is the PCR amplification that is happening right now. After this is done, we just click done. Now the sequence preparation is complete. Now we click the orange part to continue to part 5, which is DNA sequencing. Now here we will load it into the automatic sequencer, and to do this we click the orange tray. Click the DNA sequence machine to open the doors, and we click on the orange tray to load it up. Now we click on the inner door, and now there's an animation showing the sequencing. And we just click done after we have finished. Now we completed DNA sequencing and now we move on to the sequence analysis. And here there is a series of steps that we need to follow, which is step one, we need to open the output from the sequencer. Here there will be a new tab open and we'll be see a sequence. And then after that, we go to the NCBI website where we'll be finding, where we'll be using it to identify the bacterial sample. First sample D, which is a blood sample, the bacteria for this one was Salmonella typhus, and according to the NCBI, it was correct. So as I said, that we'll be using the instruction that they told us in the virtual lab, and now this is the first step, which we open this, and this is the gene sequence that we get from the sample. And now this will be adding it into the NCIB website in order to get a full identification of the bacteria. Now move on into step 2, which is to paste the sequence into the NCIB website. And what this will do is it will be able to identify the bacteria. So after waiting a couple of minutes for the identification, it has been identified as the Bartonella hensleyi bacteria. So after using the NCIB website to identify, I chose the bacteria option here, Bartonella hensleyi, and this proves it that was the identity of sample A. So as I said in the beginning, I'll be doing samples A to F, and I only recorded for sample A as it includes the same steps for the samples B to F. So by the time you're seeing here, I have done all the steps, and now it is only left to identify the bacteria, which for this stool sample, I found out it was Escherichia coli, and that is correct. For the identification of sample C, which is a urine sample, I found out, according to the NCIB website, that the bacteria is Pseudomonas aeruginosa. I do not know if I did pronounce the name right, but that is what I believe it might sound like, and it is correct. For sample D, which is a blood sample, the bacteria for this one was Salmonella typhus, and according to the NCBI, it was correct. Now for sample E, which is a sputum sample, we click on the computer screen, and according to NCIB, the identity of the bacteria was Yersinia pestis, and it was correct according to the NCIB, and it was proved by the virtual lab. And now for our final sample, which was a stool sample, after going to the NCIB website and identifying it according using the sequence that I got, I click on the computer sample and I got that it is Yersinia enterocolitica, and it was correct. And now I have successfully completed I successfully completed the lab, and also identifying the other samples, which is sample A was fluid from lymph node, which was Bartonella hensleyi. For the stool sample, sample B, the bacteria was Escherichia coli. For sample C, the urine sample, it was Pseudomonas aeruginosa. Sample D, which is a blood sample, was Salmonella typhimurium. And sample E, sputum sample, the bacterial identification was Yersinia pestis. And stool A, for the stool sample, it was the bacteria identified was Yersinia enterocolitica. And with that, I do complete my lab.